Today, we are privileged to have Kevin Pelphrey with us, speaking about his experiences as an autism dad and autism researcher. Kevin is a neuroscientist whose focus is on discovering the brain basis of autism and neurodevelopmental disorders. He is a Carbonell Family Professor and Director of the Autism and Neurodevelopmental Disorders Institute at George Washington University and Children's National Health System in Washington, D.C. and Virginia. Dr. Pelphrey is working to develop tools based on biology for both the early detection of autism and the development of individually tailored treatments. It is our extreme pleasure to have Kevin with us today, and so without further ado, please take it away.
Hi, so I'm delighted to be here. And what I thought I would do today is a combination of talking a bit about my family and my role as a father of 5 children altogether, 2 of whom are on the autism spectrum, and then talk about how that has shaped the type of science that I do, and present some research that I hope will be of interest to you, and relate that to some of what I've learned from being a parent and dad of kids with autism and kids without autism as well. So let me get started by telling you or showing you one of my favorite things to do, which is to show you pictures of my beautiful daughter when she was very, very little, up to about the age of 4 here. So let's see. Let me see if I can master using the highlighting tools. Okay, so Frances, very early in development, and she had a fairly typical birth and delivery, although there were a couple of concerns, few concerns about the rate of by which she was growing, and then her presentation as a fetus. She was kind of turned around, which led us to let her mom to have a C-section. And also there was one very scary episode of a bladder infection that her mother had while pregnant with Frances. And so that was, other than that, fairly routine labor and delivery. So Frances came into the world, very, very beautiful child, and remains so to this day. Beautiful young woman now. She's 13. So this is Frances with her very best friend for about the first 7 years of her life. Her dog, Jeremy, is an English setter, and Frances with one of her favorite toys as a child. And here is Frances. I think she was about 4 years old here. And we took her to see the Wiggles, and she might be familiar with if your parents in particular. And she was absolutely fascinated by and just obsessed with the Wiggles. And at this point, we had some pretty serious concerns about her development, not because of her obsessions with the Wiggles, but certainly the strong interest in the one category gave us a lot of concern. But what was really concerning is that at this point in her development, she was still barely talking, and we had been concerned about this for some time. And in fact, her first word was Henry. And she said, and this is Henry the octopus. Let me actually change the color. Brilliant. Okay, yeah, so that's Henry. And Henry the octopus was her favorite Wiggle. And, so we would watch this television show together, and she would sit in my lap, and she was very excited whenever Henry came on. And I would try very hard to get her to say, there's Henry, but she never would. And eventually, when we were working on my computer one day, we were looking for pictures of Henry, and she saw that you could order Henry online, and she loved plush toys, as you can see here. And so I ordered her a Henry, and Henry eventually came. I can't tell you how hard it was to find the Henry on eBay because they were already out of circulation at that point. So I think I paid quite a bit for Henry. And I was a postdoc at the time. But Henry arrived, and Frances's mom was teasing her about Henry, saying, "My Henry," and acting like she was going to take Henry. And Frances's really her first words were actually a complete sentence. She said, "My Henry, no," and grabbed it and started screaming and running around the room and having a temper tantrum about her Henry possibly being taken away. And that's kind of the way things went for quite some time about Henry, and I think she actually still has Henry, although she would be embarrassed for me to tell people that she still sometimes carries Henry around in her backpack. But Henry the octopus was a very important part of her growing up, and these kinds of strong interests in categories or types of objects and groups of people remain something very important to her, and she's gone through a number of them. And as she's gotten older, now that she's a teenage girl, for example, some of them, and she still is very open to talking about them, even with Dad. But some of them now are like Zac Efron and the male stars of High School Musical. And, she kind of categorizes them by cuteness as opposed to what category of animal they are. And it's unusual as a dad, I think, certainly from growing up and kind of watching my sister and my father interact, for her to share that with me. It's delightful at times, but then other times I don't really know what to say because I don't have an opinion about whether or not Zac Efron is the cutest boy in the world, although probably is up there in the top 5. But it's certainly, something that she thinks about quite a bit. And so she is growing up very, very rapidly, but it took us quite some time to figure out what was going on with Frances, because she was not only our first child, but she was a young girl who was having developmental delays. And ironically, I guess, ironically, or counterintuitively, when we were going to physicians and our pediatrician, the answer when we raised concerns was often, well, she's a girl, she'll grow out of it. And knowing now what I didn't know then, either as a parent or as a scientist at the time, although I kind of knew it as a scientist, but when you're a parent, what you know as a scientist, sometimes you don't really want to think about as a parent, and you're willing to accept things like she'll grow out of it because that's really what you like to hear. And so I knew, though, on some level that, well, actually, she's less likely to grow out of it because she's a girl. She should be advanced socially, if anything, and advanced in terms of language development. My PhD advisor, Steve Resnick, had written, been an author on one of the most important language assessments from MacArthur, and had normed it, and so I knew better, but I certainly was willing to hear that she'll probably grow out of it. And I think that even specialists at the time were hesitant to make the autism diagnosis because Frances seemed very social. Her choice of obsessions were always very social. And kind of the way she perseverated on them was, on the surface of it, very social. But underneath it, it was different than where she should have been relative to other girls. But I didn't really have that reference point. And certainly compared to what I knew as stereotypical boys playing, she seemed quite interested in social interaction. So it was a tough call all the way around. But eventually she received an autism diagnosis, and it was kind of a strange irony there, in a sense, which I'll get back to in a minute. So let's see. Now, I mentioned that I have several other children, 5 altogether. 3 of them are my biological children and 2 are stepchildren. And so kind of talking about my biological kids, I have 2 boys and a girl. And what you're looking at here is just some pictures of those 2 boys and my daughter. And let's see, so you've seen Francis, and this is Kenneth, and this is Lowell. And so this is Lowell, and here we go. So it was an interesting, I think, kind of unusual family situation in that our daughter very clearly had developmental problems, but our sons, so our second child, Kenneth, if anything, quite accelerated in terms of his development. And our second son, Lowell, if anything, again, seemed accelerated in terms of a lot of cognitive development. So we were looking at this, and it was intriguing. And I remember after the diagnosis, we had, which was done at the Yale Child Study Center by my colleague Pam Ventola, who's now a very close collaborator of mine. That's how we met. We had genetic testing done and a whole exome sequencing at the time, I believe, and found this duplication on 4P that Francis had, and then, I was inherited from me. And I remember kind of running this by Matt State, who remains a close colleague of mine, but we were both at the Child Study Center at the time. He's chair at San Francisco now. He said, well, it's probably, it's nothing because she inherited it from you, and you're obviously normal. And I kind of laughed, and, anyone who knows me well or knew me as a child would be very uninclined to say I'm normal. I am happy to talk at length about stuff I feel like talking about, but most days I would just as soon not engage in small talk. And certainly as a child, I didn't speak for quite some time. And my parents, my father, that was pretty normal for him from his point of view. So there's sort of this unusual, I guess what we would call now the broad autism phenotype in my family, going back quite some time. My family, my generation was the first to really be able to attend college, but my father was a mechanical genius who could fix anything mechanical without even seeing it before, and the reason I could go to college was his success in being able to do that. And, his father was an electrician very early in the development of the Appalachian Mountains and coal mining industry. So kind of we were nerds before being nerds was cool. So it's sort of, I thought it was kind of unusual that Matt thought, well, you're normal. Maybe normal relative to PhD geeks in neuroscience, but relative to the rest of the world, not so much. And then what was kind of funny about the picture that I'm drawing on now, and I guess so this one, was that at the time, I think Abha Gupta and I were, she was one of Matt's students, mentees. We were playing around with this as a family tree, and this was when Kenneth had been genetically tested, but Lowell had not. And so we knew Kenneth didn't share this genetic feature, but we didn't know about Lowell yet. And then we knew Kenneth was typical, certainly didn't meet criteria for autism. He jokes, or not so much jokes, I like to think he's joking, that he suffers from typically developing middle child syndrome. And then his youngest brother, at the time, in drawing this, I thought for all the world was typically developing. And it's funny because he was in one of Kasia Trawaska's infant sibling studies. And so he was being followed very, very carefully. And I enrolled him because she mentioned in a meeting needing more kids, and especially kids that were seeming to be typically developing. And so I thought, oh, well, here's one for you. And she called us over for to go over the evaluation at about 12 months. And my wife was there, and she said, well, where's Kevin? And, well, Kevin's over at the lab. He said to just go ahead and do it without him because he's, running a participant. And she said, well, no, he really needs to be here. And, well, he said he knows what you're going to say. And Kasia said, he really doesn't, and he needs to come over here. And I kind of made my way over. And she said, I'm concerned, and we need to start very intensive early intervention. And I was sort of like, what? And I don't get it. And she started showing me some of the things she was seeing, and it's like, oh, okay. This is nowhere near as clear as Francis, but yet because some of the things that Lowell was doing were so stereotypically autistic, but yet so subtle, and he was so cognitively advanced, I was missing it from that perspective. And so this kind of carefully trained eye is picking up on it. And so Lowell received very intensive early intervention. And it's one of those things where we'll kind of never know if we were being too careful in making the early call, or if Lowell is what we would call an optimal outcome child. But at this point, he doesn't meet criteria. He's very different from his brother. Of course, any person is going to be different from their brother, but it's like night and day in terms of personality. Lowell's very shy and introspective and a little socially anxious, whereas Kenneth tap dances, joins every play he can, and has never met a stranger and can make small talk all day. Lowell has subsequently been genetically tested and shares this 4P duplication. So it's a really kind of interesting pattern of features. And then what has kind of become interesting from the point of view of the genetic science is that over time, this particular difference has become one of the kind of known potential causes of autistic features. There's several cases of it in the literature kind of just building up over time. So it's interesting to sort of see the way the literature eventually kind of catches up with what our family experience was. And, I've been very interested in this particular duplication and very likely to participate in things like SPARC or Simon Simplex collection or pretty much any study that's out there, because of being intrigued by how, a little difference in genetics and kind of against the background of maybe of being a boy versus a girl can change and affect trajectories. It's something that I'm kind of exploring in my own thinking about different pathways, because Frances is cognitively affected by her autism, and Lowell certainly wasn't. And so it's kind of interesting to think about that and see it unfold. Okay, so now, so this is Frances as she is now, and or actually last year, and we are at our favorite restaurant, and it's our favorite restaurant because they will actually turn down the music when we go to it. One of the things that has been an issue for Frances after puberty was and actually, I'm not going to tell that story, sorry. But so after puberty, Frances developed epilepsy, and had several seizures, and they are in part seem to be brought on by lots of loud noise and the associated sensory stress and overload. So that's been a big issue for us, and it's interesting because she'll play her favorite movies and songs with the headphones that you see there as loud as she wants, and that doesn't seem to bother her. So I think it's more the inability to control what she's having to be exposed to, more than the sensory impact itself. And so, just as one of those things to think about. And when you're thinking about autism and epilepsy and their co-occurrence, there's two kind of hot spots within autism for increased risk of developing seizures and epilepsy. One is, of course, very early, and then so present more or less from birth. And then if you kind of escape that experience or that associated medical condition early on, you will statistically speaking, you will be fairly insulated from seizures until adolescence and the onset of puberty, which is another time where you will have the higher risk for developing seizures. So there's really, to summarize, there's two hot spots. One is the very early in development, and then again around puberty. And we don't fully understand all of the reasons why, but it is something to think about. And it's not, it's one of the more common associated medical conditions. We could put a number on it, about 10 to 15%, and it depends on how you're sort of defining autism, and what parts of the spectrum you're looking at in order to then look at the rates of epilepsy. So it's a complicated question. Okay, so now kind of moving into the science of what we have, of what I've been up to based on my experiences with my daughter, and now my sons as well. I'm fascinated by the sex differences that we see, and this is like 3 articles that were in Spectrum News that appeared in fairly close proximity that were just fascinating and kind of covered different aspects of sex differences and questions about diagnosis, questions that I've alluded to a little bit, and the difficulty of getting a diagnosis at the right time, whether or not autism is the same in girls and boys. Are we actually talking about 2 different disorders in addition to all the other heterogeneity that we talk about in autism? And so, I wanted to put these up to say that it's both, it's actually very exciting on the one hand, but also kind of depressing because I don't know how much progress we've made yet in answering these questions. But I'm really excited that the field seems to be very excited about these different questions. And in particular, I'm intrigued by questions around temper tantrums, so-called temper tantrums. I like to think of it as differences in emotion regulation and their relationship to autism and sex differences in abilities to emotionally regulate and autism. So that's something that our group is studying quite a bit, and that's something that's directly related to trying to understand my daughter. And, I kind of question every day, given that I have a 15-year-old typically developing daughter and a 13-year-old daughter with autism, and sort of the question of what's autism and what's a teenager and understanding teenage development. I don't know. Having a PhD in psychology doesn't help answer that question at all, even in developmental psychology. So it's intriguing to kind of think about that. And I like to think a lot about how adolescence is a period of development with incredible reorganization and the opportunity to really think about period of plasticity that's on par with very early infancy. And can we think about ways to leverage that reorganization of the brain and the plasticity to come up with some really creative interventions that will help our kids get a booster as they move into early adulthood at a particularly opportune time. So launching into a couple of things that I want to show you from our science, I want to talk about this concept of biomarker, right? Because in a way, had there been beautiful biomarkers, I could have tried it out in my daughter when we started wondering about autism and had an answer. A biomarker promises to give you a sense of whether or not a condition exists in a person and the level of the condition. And by giving you that, it tells you something about, could you use it to monitor change in that condition over time? So you all have undoubtedly heard the term biomarker in different contexts. And I thought, the real issue is that not having biomarkers leads us to not really knowing what treatments to use for which people, and that leads to wasting a lot of time and a lot of resources. And so, where we would like to be is at a place where we can say, all right, you have this condition we think of as autism, and now we want to think about how to measure, that and understand it unfolding over time, and then develop a particular treatment that's targeting that mechanism. To do that, we need the biomarkers. And so, but what are we really talking about? So I laid out here some criteria that I thought would be good for evaluating biomarkers, as many of us in the field are trying to develop these. And I wanted to put up this little cartoon from a Nature article that I was reading a couple years ago that was titled Imprecision Medicine. So you've probably all heard the term precision medicine, imprecision medicine. And this was beautiful for putting into context some of our aspirations. So this is a listing of drugs, none of which are for autism, but all of which are kind of blockbuster drugs, real success stories in the field. And they were, made it through the FDA process, right? So here we've got the number of people that you have to give the drug to in red before you get a responder in blue. And that was kind of stunning to me, because I think about, other areas of medicine with a great deal of envy, and I think, oh, well, they've really got it worked out. And I can't tell you how many grant proposals I've written where I talk about, oh, if only we were like heart disease and we could measure cholesterol, and then we had something that affected it, that thing, and we could measure its reduction. And, well, there you go. There's Crestor, and this is the number of people you have to give it to. And by the way, it has quite a high profile of side effects. People you give it to before you get one that it works in. That's kind of that's stunning to me. And so I don't know. Maybe we're not doing so bad in the field of child psychology and child psychiatry. But let's talk a little bit about these criteria that I put over here that hopefully you've had a chance to digest. I won't read them to you. kind of coming back to the story of my family and myself. So I, as a young scientist, I thought autism was really impossible to make progress in. And I was working with Joe Piven, and he had been working on this for years, and progress was slow, not because of Joe. He was making great progress relative to what one could make, but the questions were giant. And this was pre-Simons Foundation being involved. Things just looked bleak. But I was doing work that other areas of basic science were really excited about, trying to figure out how the brain processes social information. So, like, how do you understand other people? So we were discovering the parts of the brain, like this little guy, the posterior superior temporal sulcus, that understands other people's motions and what they mean. And I was getting papers published, and in academia, that means I was getting job offers, and they were telling me, if you want to succeed and get tenure and be able to support that new family you have, at the time, my young daughter, who was a newborn, and we had, no real worries at that point about our daughter's development. You really need to move away from autism and move towards basic cognitive neuroscience. And so I did. And then I kept doing my work that was relevant to autism, but I tried very hard not to study autism at all. And, that expression, every time you try to get away, they pull you back in. That's kind of what it felt like when we got the diagnosis, because the day after we got the diagnosis, and I was still in shock, I had moved to Carnegie Mellon and had become an associate professor there in their psychology department. And I was planning on, living a happy life of teaching cognitive psychology. I was very happy with my daughter, and she was doing well, but struggling, and she was going to the children's school there, which was amazing. But then when we got the diagnosis, I thought, wow, I just moved from Chapel Hill, North Carolina, where I had access to the world's greatest experts. And, now I'm in Pittsburgh, and I'm not really sure what to do. So the next day I got a call from Ami Klen and Fred Volkmar at the Yale Child Study Center, and they said, we've been following your cognitive neuroscience research, and we'd like you to move to the Yale Child Study Center. And so probably I was the only candidate that they called that cried, and I tried not to accept the job before they offered it to me, but I made it clear that I might be interested in the job, and I mentioned Francis. And they kind of paused, and I heard them kind of whisper, and then Ami said, well, just bring Francis, and let's worry about the job interview later. And so, they saw Francis and Pam Ventola, who I mentioned, and by the end of it, I had worked out the details with Fred, and we were moving the next month to set up the lab at the Child Study Center. And that was primarily in order to be able to develop the work that I wanted to develop in the context of Francis and children like Francis. And so to kind of accelerate the story a little bit, we took this basic science, this kind of understanding, starting way back in 2003, about understanding basic brain development and how people understand other people, and started applying that to autism, doing things like trying to understand, could we use it as a biomarker? For example, if we find a group difference in a discovery group of kids without and kids with autism, can we apply it to a new replication group and be able to say, this person has autism, this person doesn't, using, for example, just the brain scans? And an answer was, well, yes, sort of, but only for boys. So this was another kind of shocking event. When I saw this data, I saw the group data combining boys and girls, and it showed that some of my favorite regions from my basic science career were nicely predicting an autism diagnosis, but only for boys. That's what this graph means. For girls, we were breaking math. What do I mean by that? We were doing worse than chance in predicting autism in girls. That can only be true if we broke math, math doesn't break, or if boys and girls with autism are two different populations. So I'll pause there for a minute, mostly because I'm talking to my computer screen. And normally I kind of look at the audience and hopefully people would be thinking, that's strange. So all of these kids have had an ADOS and an ADI and have been seen by some of the very best clinicians in the world and made it through all of those diagnostic hoops. They are more similar than you could possibly get by chance because they've made it through all of those similarity-inducing devices, and they've been given this autism diagnosis. Then we put them in a brain scanner, and we show that even though their behavior has similarities that leads you to make a diagnostic call, the underlying brain differences are different. And so that gave us great pause because everything we had been publishing on didn't take that into account at all. And so then we realized we really ought to be studying girls separately. And so that's this network that we've been running, this NIH Autism Center of Excellence. And so we have multiple sites shown here on the East Coast and the West Coast where we're getting as many girls and boys with autism as possible, as well as their parents, and doing brain imaging and genetics, really great phenotyping biomarkers on these kids. And now we're following them over time as they enter into young adulthood. And I just wanted to show you one thing from that. If you simply look at the patterns of resting state activity and you say what most kind of differentiates girls with autism from typically developing girls, and then boys with autism from typically developing boys, and you say that network activity is our biomarker, then you think, all right, well, what do the networks do? And you go into this database called neuroscientist, and you compare the maps you're getting, the networks you're getting, to all of the published ones that are housed there. And you say, tell me what my maps look like with relative to all the other literature for girls and then for boys. And then I simply made these little wordles based on the correlation between the terms that came up, the psychological terms, and the underlying network. So this is the network, what it's doing correlated with the term, all right? For boys, the things that differentiate them are person perception systems, social perception systems, language systems. For girls with autism, relative to typically developing girls, the brain systems that are most different at rest are really more about language comprehension, anxiety, and emotion regulation, with a whole bunch of attention thrown in there as well, and very curiously, quite a bit of tactile perception and sensory perception. And so what this is saying, and what might be the most important potential for a biomarker in autism, is to give you new leads about the underlying mechanisms of the disorder, as opposed to simply giving you a stand-in for the diagnosis. Because if these girls and boys have already been made as similar as they possibly can by virtue of going through a diagnostic process, I'm asking you to think of what if. What if that diagnostic process actually masks or gets rid of, throws away a lot of heterogeneity, and instead puts people into yes or no diagnostic categories that then make them artificially more similar. But really there's a girl autism and then there's a boy autism. And when you look at the underlying brain network, you get these robust, statistically meaningful differences that say that different brain networks are involved. If that's true, then you would want different treatment, because you would need treatments that address this and these brain networks, these causes, and the treatments that address these brain networks, these causes. You wouldn't want to use the same treatments for girls and boys. Now, how about prediction? I think this is really the new frontier for biomarkers. So in autism, this has been done in other neurodevelopmental disorders, neuropsychiatric disorders. This is Pam Ventola, who I mentioned. This is her work, her baby. And this is work that she's been doing around pivotal response training, which is a form of behavioral intervention that's evidence-based. She's been doing this beautiful work where she's doing 16-week intervention with these kids. And then we're scanning them before and after. And so we're able to look at their behavioral response to PRT, pivotal response treatment. And then we're also able to look at their brain responses. Now, the behavioral responses, we're using currently the social responsiveness scale as an outcome measure because we want a behavioral measure to say whether they're improving. What this is showing you, this red line here, is the average response. This is just two different ways of looking at it. And as you can see, on average, the kids are doing better. Their symptoms are going down over time from pre to post, okay? But each little black line is an individual child, and what you see is lots of heterogeneity. And this happens in every treatment study. Kind of what you're wishing for if you're developing a treatment is an overall change. But some kids are going to get a little worse. A lot of kids are not going to change at all. And quite a few kids get quite a bit better. And we already knew this. We just needed to prove that we could also do pivotal response training. It wasn't just a West Coast thing. And so we did that. But more importantly, we took brain scans of these kids before and after, as I mentioned. And then we were able to use those scans to predict which kids would benefit more and less from the treatment. So why would we want to do that? Well, this treatment, if we weren't giving it away as part of the research, would cost about $30,000 to do. And in a lot of states, insurance doesn't cover this. And if we don't know for whom it will work, then that's potentially a lot of wasted time and money. It's also very time-consuming. Parents have to drive the kids to the location. It's quite a few hours per week. There's a lot of home visits. One parent almost has to stop working, and sometimes there's only one parent there. So if there's no parent working, how is this going to get done? So it's important to be able, in terms of precision treatments in autism, to be able to make these types of predictions. But it's also important because with brain imaging, in contrast to just about every other biomarker technique out there, there's both a where and a why to it. So when I show you a diagram that says this spot in the brain, the posterior STS, this circuit in the attention network, this circuit around the insula and ventrolateral prefrontal cortex involved in emotion regulation, and this circuit right in the middle of the brain's reward system strongly predict success in PRT. I'm making a mathematical prediction, but I'm also saying literally that if you made these spots in the brain more active and more interconnected somehow before the child or while the child is getting PRT, they will do better with the PRT. It makes that type of strong prediction. And because it gives you a where in the brain as opposed to just a when in the brain, like, for example, EEG, you know what neurotransmitters are expressed there. We know from genetic data what genes are expressed there and when. So we know what molecular pathways to pursue. So, for example, these brain regions scream something like dopamine and oxytocin. And so, in fact, when you give intranasal oxytocin, those same brain systems, just with a single dose, about 45 minutes later, start to activate in young children with autism in response to the same type of socially intriguing stimuli. So now we're following, as part of Simon's Foundation funded work, we're going to be following kids with PRT but giving them intranasal oxytocin or placebo and determining whether or not they do better with treatment if they were a bad candidate for PRT, if we give them oxytocin to turn them into a better candidate. And so we're very, very excited about that because it really gets us to the point of talking precision intervention, not just biomarker to observe and predict, but biomarker to begin to intervene and use as a lever for treatment and intervention. Okay. Now, people often say to me at this point, and this is looping back to my very wild family, well, okay, that's great. And, you know, in FMRI, I know a little bit about that. Big MRI machine. It's not going to be in every doctor's office. You know, how are you going to do that? It's not practical. You know, every child with autism, you can't give them an FMRI prior to, you know, starting an intervention. You're just going to make the intervention more expensive. Like, okay, yeah, I get that. It's sort of like this. You know, I say to my amazing wife, Annie, you know, I want a Ferrari. And by the way, in fairness to her, she's the one that would want the Ferrari. She loves Ferraris. But she reminds me that this is our situation, and we're very happy about our situation of these 5 crazy kids. So maybe something like this, which is a very cool Vanagon, but it's not a Ferrari, you know, but I can still maintain my dignity because it's not quite a minivan, even though, yes, I guess technically it's a minivan. So my point is, all right, we've got techniques, you know, MRI is great, and we can do that. But we have other techniques like EEG and optical imaging and eye tracking that we can use as proxies once we really understand how they relate to the gold standard, which is MRI and FMRI. We can then begin to use those as biomarkers as stand-ins for that more mechanistic understanding. So that's, so, you know, that's kind of my answer to the practicality question. But I'll also say, you know, I don't know. MRI is an FDA-approved device in every hospital in the United States, and the pediatric guidelines state that if a child's diagnosed with autism, they should get an MRI. And so I don't know that it's as crazy as buying a Ferrari when you have 5 kids. So maybe it's not so impractical after all. So I want to stop there in terms of the science and open it up for questions to you. I want to make sure I acknowledge different people who funded this work. And also, you know, I mentioned Pam Ventola several times, but many other colleagues, Mike Crowley and others who were there doing this work with me. And okay, and so now let's see. So I'll move on to questions.
Thank you so much. We got a few questions in, but if others have any, feel free to send them through the chat window. So I think someone is asking about, out of the different methods you showed in your previous slide before the acknowledgement, what would give a detailed autism report? So someone had a report done in 2008, but now their son is older. So would one of these be advised?
So all three of the techniques that I'm showing here are different types of brain imaging techniques, essentially. For a report, you want a multidisciplinary assessment that, gets you a psychologist meeting with your child psychiatrist, speech language person, job coach, occupational therapy, and then maybe an MRI or an EEG, especially if there are concerns about potential seizures, would be indicated. But that would just be one part of a multidisciplinary assessment.
Okay. So you talked a lot about biomarkers. How will this affect the creation of medication and treatments that will be effective at decreasing behaviors?
Yep. So all of the biomarker attempts out there are essentially attempts at trying to develop something that gives you both a quantitative assessment of qualitative behavioral differences that you can then track over time that's reliable, and hopefully gives you insight into an underlying mechanism or set of mechanisms, so that on the one hand, regardless of the treatment you develop or try to develop, you have something to measure the disorder or the symptoms that's reliable. And then on the other hand, it tells you where to be looking as you're developing the treatment. It gives you insight into potential causes.
And someone is asking if oxytocin is in clinical trials for autism.
Yeah, there have been several that have been done, and then there's one that's ongoing. The results should come out pretty soon. I'm just scanning the list of people that are online right now to see if any of them are here, because they'll be able to tell much better than I, maybe Gabriel. But it's run by Lynn Sickich. It's the SOARS-B project, and people will correct me if I'm wrong, I hope. I believe that's the name of it. And that's the largest trial that I know of that's being done very, very well, and will have quite important results coming out. And so I don't know what the early results are or anything about it, other than that it's a really well-designed study. And then I did see somebody ask, can we buy oxytocin? If you Google oxytocin, you will be taken to all kinds of sketchy sites to buy oxytocin. Please don't do it. If it ever had any oxytocin in the vial that might get sent to your house in the little bottle, by the time it gets to your house, it won't have any in there. It has to be kept on dry ice most of the time. You have no idea what's in it, really, and there's all kinds of scams running, unfortunately, because of people like me who have published on autism and oxytocin. And even though we've said very clearly it's too early to be doing anything with this, people do, try it, so please don't. There was a time when you could buy it on Amazon. I don't know if it's still there. Really unwise, and at best, it's a waste of your money, because we don't know anything about it. We don't know what we need to know about it yet. It's interesting science. It's a potential strong lead, but we don't know what we need to know before people are buying it and trying it.
So
sorry.
What are your thoughts about the commonly reported 4-to-1 boy-to-girl ratio in autism?
My thoughts on it.
I know that's a big question.
I have a feeling. I think probably the kind of what the person asking the question has in mind is, do I think it's closer to 1-to-1 or smaller ratio? I think that there's a group of people in the world that we're missing from this category of autism that is would make the ratio closer to 1-to-1. I think, like every condition, women are the stronger sex on just about every condition. The rare examples are eating disorders, depression, and anxiety, two that come to mind quickly. But otherwise, so I do think there is a bias and kind of a female protective effect, but I do think we're missing a group of girls that have the brain characteristics, have this vulnerability, but because of the trajectory of female development, don't express autism in the way that our measures capture it, but it puts them at risk for other conditions. And I have a kind of a pet theory, a theory that I think about a lot and kind of wrestle with in my head, where there's a very important connection between eating disorders and autism, and at the intersection of girls, and that what kind of causes eating disorders in girls, had they been a boy, they might have had a much more classic presentation of autism. And I kind of work on that in my, and I haven't really written that out. I probably need to. But even if it's just to do away with it as an idea. But it's something that I kind of play around with in my head. And it's funny because just, since I'm talking a lot about my kids, neither of my daughters has any kind of eating disorder, other than I'm impressed because they can eat anything and like not gain an ounce. But it's amazing. But there's this kind of, what has always struck me is Frances, what she likes to eat and her food choices, and anyway. But I'm kind of belaboring the question. But the question was a simple one, which is, what do you think about that? And I think that we're missing a group, and if we could be omniscient somehow, there's this group in multivariate space that we're missing. And if we define disorders differently, kind of a brain-based taxonomy instead of a behavior-based taxonomy, we would see them. And I know that's kind of sounds weird because, well, we kind of only care about behavior, but not me. I care about the behavior of the central nervous system, not just overt behavior.